Hi, this is Josh, Dream Body Clinic, and today we're going to talk about heart health. So we're now working with Muse cells. These are true pluripotent stem cells that were discovered back in 2010 by Dr. Dezawa. So at the end of this video, you're going to see Dr. Dezawa herself explain how these work and show a video in real time with Muse cells that were activated with a dye and administered to mice with heart issues. They'd induced heart attacks into them and then administered the Muse cells. And you're actually going to see the Muse cell go to the damaged area of the heart. It starts eating the debris of the bad cardiomyocytes, which are the heart cells, and then turn into new cardiomyocytes. So Muse cells will go to damaged tissue, start eating up the debris in the bad cells, and then turn into those types of cells. It's really incredible. So they are a true pluripotent stem cell, meaning they can turn into any of the cells from the 3 germ layers. So basically any cell in your body, but they only seem to do it with areas that are damaged. They're smart. They're non-tumorigenic, so you don't have to worry about cancer or anything, but they'll go in, and in this case, you're going to see them turn into new heart cells in real time under the microscope. It's incredible. So we're now offering Muse cells for humans. You can do these now. We've technically been offering for over 8 years because they're found within mesenchymal stem cells, and we've been doing those. But now we can isolate the Muse cells and replicate them and administer them. In fact, I did 20 million yesterday. So those are flowing through me right now, and they're really cool. You can learn more about them at www.dbcmusecells.com. Now stay tuned to watch Dr. Dezawa show the video and explain this in her own terms.
I would like to show you the video in the next slide, but before that, I would like to explain the background of the experiment. Now, we prepared human Muse cells that were introduced with the Gadafor imagery. So when Muse cells start to differentiate into cardiomyocyte, they express imagery red fluorescence, right? The Gadafor is the first transcription marker that start to differentiate for the cardiomyocyte lineage. Those human cells were supplied with GFP mouse cardiomyocyte fragment treated with the anti-cancer drug etoposide. So they will make the fragment from the cardiomyocyte, the green cells, and they were supplied. Okay, so those translucent cells were human Muse cells, and they were incorporated with the Gadafor imagery, and they were supplied with the green dot. This is mouse cardiac cell fragment. Okay, please pay attention to the central, the one single cell there. Okay, so this is 2 days live video. So the cell captures that fragment, they eat it and digest it, and by next day they already start to express Gadafor. So they are working towards the cardiac lineage. By 2 days, the round cell shape changed to longitudinal with a striated-like structures you can see here.
Yeah.
Right? It's more like cardiomyocyte, right? And then they're, turned into red. I think I can repeat this. It's very quick. It's happening in a days.